In 2006, a researcher in Kyoto took a skin cell from a grown mouse. An ordinary cell. Finished. Specialized. It had one job for the rest of its life, and every textbook agreed it was never going to do anything else. He added 4 proteins, and the cell forgot. Not damaged, not killed. It went backward, back toward the kind of cell it had been at the very beginning, before it was anything in particular. Biology had a rule that development runs one direction, like a marble down a hill. Shinya Yamanaka rolled the marble back up. He didn't edit its DNA to do it. Not one letter changed. Your DNA's the hardware. What he rewrote was the software running on top. And until that moment, nobody was sure that software could be rewritten at all. 6 years later, he'd have a Nobel Prize. 20 years later, this June, the same idea entered its first human trial, in a living eye. Tonight's a showdown. Full strength, both sides, out loud. I'll make the best case I can that this points somewhere. Then I'll hand the skeptic the microphone and let their case stand. No thumb on the scale. So let's start where it landed, and work backward to how we got there. In January 2026, Life Biosciences received FDA clearance to begin the first human trial of what's called epigenetic reprogramming. On June 9th, the first participant was dosed. The company was co-founded by Harvard's David Sinclair. We'll unpack that phrase properly in a minute. The targets are 2 age-related eye conditions, glaucoma, and a rarer one called NAION. Both are the kind of diagnosis that quietly ends careers and driver's licenses. And the goal isn't to replace those damaged cells. It's to reset age-related patterns inside them, without erasing what those cells are. Why an eye, and why now? Sinclair's lab spent years asking why some cells age while others, in the same body, barely seem to. In 2020, they published results in Nature. They'd restored vision in mice after optic nerve damage, using an early version of the technique that's now headed into people. An eye's also a good first test because you can watch it work, or fail, without opening anyone up. They're using 3 of what are called the Yamanaka factors. Not 4. On purpose. That detail matters far more than it sounds, and we'll come back to it. So let's go back to where we left off 2 episodes ago, in The Cell is Running Code. We showed you the letters, A, T, C, and G. 4 of them, read 3 at a time. They spell out proteins the way letters spell out words. Francis Crick called that arrangement a frozen accident. He meant the mapping isn't forced by chemistry. It's closer to a convention, like letters standing in for sounds. We argued a real working code like that needs a source. Then we conceded the obvious objection completely. Evolution demonstrably writes new information. Once a working code already exists. We didn't contest that. We won't tonight either. But here's what that episode didn't get to, because it wasn't finished yet. A code sitting still doesn't build a body. Every cell in you carries identical instructions. Skin, heart, neuron, same 4-letter book, cover to cover, no pages missing. So something else decides which pages get read, in which cell, at which moment of your life. That's the software. And we can date almost to the year when biology first found words for it. In 1962, a scientist named John Gurdon ran an experiment his own field expected to fail. He took the nucleus, the cell's control center, where the DNA lives, out of a specialized intestinal cell from a feeding tadpole. A finished cell with one narrow job. He put that nucleus into a frog egg whose own had been removed. It grew into a normal, swimming tadpole. There's a human detail here worth knowing. Gurdon's school report, years earlier, had told him that becoming a scientist would be a waste of everyone's time. He kept it framed on his office wall for his whole career. Doubt, it turns out, is a survivable starting position. That's a theme this channel keeps running into. His result broke an assumption biologists had held for decades. They'd assumed a cell, once specialized, permanently loses or damages the instructions it stops using. A gut cell doesn't need the genes for building an eye, so surely those get discarded. Gurdon proved otherwise. The full instruction set is still sitting there, intact, in every cell. Nothing was deleted. It was switched off almost everywhere, except the one page that cell needed. That single result laid the foundation under everything after it. A sheep named Dolly. and eventually a lab in Kyoto. He shared the 2012 Nobel Prize for it, with the man who's about to become the center of this episode. So if the code was never the limit, what is? In 1957, a biologist named Conrad Waddington gave the answer a picture. This is decades before the chemistry existed to explain it. Development, he said, is a marble rolling down a hill full of branching valleys. Every cell starts at the top with every option open. As it rolls, it's committing. Left groove, right groove. Once it's settled into a channel, it doesn't roll back up. Skin stays skin. Heart stays heart. That was the whole point of the picture. The hill only runs one way. We now know what those grooves are, chemically, and it's simpler than it sounds. Small chemical tags, think highlighter marks, get added onto the DNA and onto the proteins it's wound around. Add a tag here, that stretch of code goes quiet. Remove one there, a different stretch lights up. That whole layer of tags is called the epigenome. Epi is Greek for on top of, so on top of the genome. Same book in every cell of your body. Different bookmarks. Different passages highlighted, cell by cell. Now here's the question that turned Waddington's picture into something you could operate. If a cell's identity is really just a set of highlighted passages, could you erase the highlighting? Could you hand a finished cell back its blank page and push that marble back up a hill that isn't supposed to run in reverse? Now we can come back to Yamanaka and to how he actually did it, because the method matters as much as the result. There was nothing elegant about it. He started with 24 genes already known to matter in embryonic stem cells. Then he tested combinations on adult mouse skin cells, one batch at a time, pulling genes out to see what he could lose. 24 became a handful. The handful became 4. Shinya Yamanaka and Kazutoshi Takahashi published it in the journal Cell. 4 transcription factors, proteins whose entire job is switching other genes on or off, delivered together into a grown skin cell. That's enough to send it back toward what it was on day 1. Every highlight erased. The marble back at the top of Waddington's Hill. A human version followed the next year, and it's why he shared that Nobel with Gurdon. Sit with the strangeness of that for a second. 4 proteins. Not a machine, not a surgical instrument. 4 molecules the cell already knew how to make, supplied in the right amounts and the right order. And a 50-year-old assumption about what cells can do stopped being true. So that's the case. A second information system layered over the first that we can now deliberately operate. And before I hand the microphone over, I want to show you that this layer isn't only a laboratory curiosity. It's been leaving fingerprints on human beings for a very long time. We just couldn't read them until recently. In the winter of 1944, the Netherlands starved. The Nazi occupation had cut off food transport, and for about 6 months rations in the western cities collapsed to famine levels. Then, in May, the war ended, and food returned almost overnight. It's a horrifying piece of history. It also created something no ethical scientist could ever design on purpose. A precisely dated window of severe deprivation in a whole population, with meticulous Dutch birth records to match. Decades later, researchers went looking for the children who'd been conceived during those months. In 2008, a team published their findings in the Proceedings of the National Academy of Sciences. They compared people conceived during the famine against their own brothers and sisters. Same parents, same household, same genes to a first approximation. 6 decades on, the famine-conceived siblings carried measurably different chemical tagging on a growth-related gene called IGF-2. 6 decades! From a few months of hunger before they were even born. And here's the part that matters for tonight. Their DNA sequence hadn't changed, not one letter. What changed was which parts of it were switched on and how loudly, and that difference outlived the famine by 60 years. That's the epigenome doing what it does. Recording, adjusting, persisting. You can watch the same thing in identical twins, without the tragedy. 2 people, 1 genome, copied letter for letter, and yet they drift. One develops a condition the other never does. Their chemical tagging patterns, close to matching in childhood, separate measurably over a lifetime of different food, stress, sleep, and air. Same book. The reading diverges. And notice what that means. If your DNA alone determined you, identical twins would stay identical. They don't. So something else is doing real work, and that something is what the next 10 minutes are about. Now I owe you the honest objection, at full strength, because it's a good one and it doesn't need my help. There are 4 parts to it, and I want to give you all 4 before I respond to any of them. First, nothing about this mechanism is exotic. Every enzyme that adds or removes one of those chemical tags is an ordinary protein, made from an ordinary gene, sitting in the genome the entire time. It's chemistry doing what chemistry does. Second, this regulatory layer isn't special to humans, or even to animals. Some version of it shows up across nearly all life that has a nucleus. Plants, fungi, single-celled organisms. That aligns well with what you'd expect if it evolved gradually and early. Perhaps not what you'd expect from something bolted on afterward by a designer. Third, Yamanaka's 4 transcription factors aren't secret switches hidden for someone to find. They're master regulators the genome already had, doing a job it already does during ordinary embryonic development. He didn't add information to the system. He turned up 4 dials that were already on the panel, at levels the cell already knew how to read. And fourth, this is the one that cuts closest to what I'm doing here tonight. Human beings are pattern-matching machines. We see faces in clouds and intention in static. A layered, information-controlling system looking designed might say more about how your brain hunts for minds than it says about the biology. That's not a cheap shot. It's a real caution. And it applies to me at least as much as to anyone watching. I'll concede all 4. Not most of them, all of them. So let's leave that standing, because it isn't going anywhere, and I don't want to rush past it. Let me show you something else instead. ER100 isn't a lone headline. This is a field now, and the money and the researchers make that plain. In January of 2022, a company called Altos Labs launched with $3 billion behind it. Investors included Jeff Bezos. It's built around exactly this idea, partial reprogramming, to reverse cellular aging. And its scientific advisors? They include Shinya Yamanaka himself. That's the same researcher whose discovery we just walked through, and he's now backing labs built on his own finding, nearly 20 years later. Peter Diamandis, who founded the X Prize, co-founded a longevity company called Fountain Life. It raised $18 million in fresh funding last year. He's personally invested in more than 100 biotech and health tech companies chasing versions of this same question. This isn't a handful of eccentric billionaires with a hobby anymore. It's an industry. And that's worth pausing on, because it tells you something the science alone doesn't. Money's a lagging indicator of confidence. But it's a real one. $3 billion doesn't move toward a mechanism people think is a dead end. What that capital's betting on is that the epigenome is reachable. Not just a layer we can describe, one we can get into and change on purpose in a living body without breaking it. That's a very different claim from anything on offer in the 1960s. And it's why this stopped being a curiosity and turned into a race. I want to be careful here, though, because this is exactly where a channel like this could get sloppy. A lot of money chasing something doesn't make the something true. Plenty of well-funded science has gone nowhere, and longevity research in particular has a long history of overpromising. So I'm not offering the funding as evidence for the argument. I'm offering it as evidence that the mechanism is real enough for serious people to bet careers on. That's all. The argument still has to stand on its own, and we haven't gotten to it yet. And the approaches aren't even the same tool. That's the part I find genuinely striking. Researchers at Cambridge's Babraham Institute worked with skin cells from middle-aged donors, including 2 people who were 53. They used a brief, controlled dose of the Yamanaka approach. The cells moved faster in a wound healing test and produced more collagen, while molecular clocks read roughly 3 decades younger. Then there's Calico, that's Google's longevity research lab, and they teamed up with a smaller biotech called Revol Pharmaceuticals on something completely different. They searched AlphaFold predicted protein structures, then used directed evolution across hundreds of millions of variants. The result was an engineered enzyme built to remove a damage marker that accumulates in tissue with age. In donated older human skin, it cut that marker by more than half, below the level measured in 31-year-old skin. That story ran on a widely watched tech podcast's science segment. One of the hosts did the math out loud, right there on the show. He priced the first commercial version, probably a cream, somewhere north of $1 trillion. Nobody in that room was underselling it. And I should separate one thing out, because the field blurs it constantly. CRISPR gets mentioned in the same breath as all of this. It shouldn't be. CRISPR edits the DNA sequence itself, the hardware, the letters. Everything else tonight leaves those letters untouched and works the layer above. Two different problems. So, now back to that detail I promised you. ER100 doesn't use all 4 Yamanaka factors. It deliberately drops the 4th, called Mike. Push reprogramming all the way, all 4 keys, and you don't just erase the highlighting. You can erase the book's identity. A cell reset that far forgets what kind of cell it's supposed to be. In a dish, that's a curiosity. In a living body, it's the thing researchers fear most. A cell that's forgotten what it is and keeps dividing anyway. Partial reprogramming, 3 keys instead of 4, is designed to reset age-related patterns while keeping the identity. And that distinction, how much to erase, how much to protect, isn't written anywhere in the chemistry. The chemistry runs exactly as far as you point it, in either direction, no further. Something has to decide where far enough ends. So here's the actual question. And it isn't, how does a cell manage its own epigenome? That's answered. Textbook. I'm not smuggling in a mystery through a door marked unsolved. The real question's architectural. It's the same shape we asked about the code itself, one layer further up. Not how does this work, but what kind of thing usually has this shape? A system that stores symbolic information is one thing. We made that case about the DNA sequence in week 4. But a second regulatory layer, operating over the first, that decides which parts get read and when, and that can be deliberately dialed to preserve identity while resetting damage. That's a different kind of thing. And it's the second time in 2 episodes we've found this shape inside a single cell. Fine-tuning worked the same way, incidentally. Not one improbable constant, a cluster of them together. That's what made the pattern worth noticing. Everywhere else we found this pairing, it was built by a mind. A symbol system plus a separate system governing access to it. An operating system deciding which programs run. A musical score's performance markings layered over the notes, telling a different reader what to do with the same page. An index deciding which chapters open first. We're not asking whether chemistry can do this. It plainly can. We just watched it happen twice, once in a frog, once in a dish in Kyoto. We're asking which starting assumption expects, going in, that a second controllable layer of access would exist at all, rather than the first code just running unsupervised. That doesn't crush the naturalist's case. And that 4th objection stays exactly as fair as it was 10 minutes ago. What it does is raise the posterior. That's the odds you'd assign after looking at the evidence, rather than the ones you walked in holding. And a raised posterior isn't proof. I won't pretend otherwise. That's the whole claim, and that's as far as I'll take it. So let me do the thing we always do here, and tell you what weaken it. Suppose someone builds a layered control system like this from plain chemistry, with nothing steering it, start to finish, in a lab. Then my argument's in real trouble. That's not hypothetical either. Origin of life researchers are working that exact problem right now. If they get there, I'll say so on this channel. Here's the other one. Suppose any sufficiently complex chemical network just grows regulatory layers on its own, automatically, wherever you find one. Then the pattern I'm pointing at isn't special at all. It's only what complexity does. I don't think that's where the evidence sits today. But I've told you what'd move me. I'd rather you hold me to that than take my word for any of this. Here's the detail I can't stop turning over. And I want to be clear, this next part is a faith reflection, not a scientific claim. Partial reprogramming keeps a cell's identity while resetting what's damaged. Not a different cell, the same one, renewed. Paul wrote something close to that shape, 2,000 years before anyone had the chemistry to describe it. If anyone is in Christ, he's a new creation, not a different person, a renewed one. A psalm gets there from another direction entirely, fearfully and wonderfully made, the writer says. That reads less like poetry the longer you sit with it. More like a lab note. Given what your cells are doing right now, just to keep you exactly you. And Colossians names the deeper pattern behind tonight's whole episode. In him, all things hold together. Not a code written once and abandoned to run. Something held, actively, the whole way through. So here's where I actually land. Say your identity survives having its entire regulatory layer wiped and rewritten. Say you're still you underneath the reset. Then what was the you that survived it? The code? The layer above it? Or something both were built to serve? I don't think tonight answers that. I think that's the next honest question. Subscribe, and let's go find out together.